Hi, I'm Dan Bean, Director of the Palisade Insectary. I'm going to describe one of our projects that can potentially enhance the implementation success and efficacy of weed biological control agents. It is the use of semiochemicals to manipulate biocontrol agents in the field, also known as beetle herding. Semiochemicals are signaling compounds produced by one organism that affect the behavior of others. Semiochemicals have been widely used in agriculture for monitoring pest populations. We use pheromones to monitor for peach moth in the Palisade area, which provides pest density information for our peach moth biocontrol program. Here we show a trap for the peach moth baited with pheromone. Semiochemicals can also be used to disrupt normal behavior patterns related to mating in pest organisms. In spite of their long history of use in agriculture, semiochemicals have not been widely used in biocontrol. The release of the tamarisk beetle Diarabda carenulata in 2001 offered an opportunity to investigate potential uses of semiochemicals in a weed biocontrol program. In 2003, after Diarabda populations began to build in western Nevada, we had the opportunity to observe swarming and dispersal behavior in the field, seen for the first time in North America. Clouds of reproductive adult beetles would hover over the tamarisk canopy in the late afternoon and colonize new trees by nightfall. Here they reflect the late afternoon sun. After the swarming flights, beetles formed large aggregations and could be found the next morning covering the tamarisk shrubs with mating pairs, as seen in the photos below. Allard Cossé and Bob Bartelt discovered that males emitted a blend of two compounds, which attracted both males and females, and this chemically mediated communication initiated aggregation behavior. Since aggregation was a critical step in defoliation, we became interested in it from the standpoint of enhancing biocontrol efficacy. Bartelt and Cossé also discovered that beetles were attracted to green leaf volatiles, a group of compounds commonly released from injured plants. GLVs are also detectable by the human nose. They have that distinctive aroma of cut grass. The GLVs were particularly attractive to beetles in the spring, after they had emerged from overwintering, and before they had heavily fed. GLV lures are made in plastic vials, and once the lids are punctured by a heavy gauge needle, beetles are attracted to the lures and end up on the sticky card. You can see the results of releasing GLVs as sticky cards become covered with adult beetles flying in toward GLVs. While the pheromone blend was not commercially available and needed to be synthesized, the GLVs are available off the shelf from chemical supply companies. The black plastic vial seen in the photo was formulated with GLVs at the Palisade Insectary and used in the field by our cooperators. While working in western Nevada, we observed that adult Diarabda would not aggregate on trees with an abundance of feeding larvae, and we hypothesized that adult beetles were able to assess the density of feeding larvae through a semiochemical signal, and avoided those trees that were heavily infested and destined to be quickly defoliated. It's clear that eggs oviposited on such a tree would not have sufficient resources to survive to last stage larvae, since those trees would be defoliated by the pre-existing larvae. Alex Gafke, working in the Weaver Lab at Montana State University, used behavioral arenas to test compounds for repellent activity. The candidate compounds are introduced into the arena through airstreams marked by red arrows, while control streams marked by blue arrows are simply air. He found a promising compound produced by damaged tamarisk, damaged by feeding Diarabda, which repelled adult beetles away from the test airstreams. The oxohexanol compound was shown to repel beetles in the field and became our first identified repellent. As a result of the foundational work of Cossé, Bartelt, and Gafke, we have a suite of semiochemicals ready for use in the field. The pheromone blend brings about aggregations of reproductive beetles. The GLVs can call in beetles particularly early in the season, and the repellent can inhibit aggregations in the event that defoliation is not the desired outcome. The photos show the glassware used to collect volatile compounds from feeding beetles, aggregations of beetles on tamarisk in the field, and the striking demarcation between a stand of defoliated plants and a green tamarisk shrub. Alex Gafke showed that semiochemicals could be used to concentrate beetles on marked trees, and he also showed that defoliation brought about by these aggregations had a negative impact on tamarisk size and foliage density. His work has clearly demonstrated that semiochemical baits can bring about a decline in tamarisk. There are a number of uses and possible uses for Diarabda semiochemicals in Tamarisk Biocontrol. We have used pheromone formulations to assess the density and expansion of populations in the field, particularly along the vast and remote river drainages of the western U.S. Tom Dudley and his group at UC Santa Barbara used pheromone formulations to track the initial dispersal of Diarabda carenulata along the lower Colorado River, showing the appearance of beetles before they were detectable using sweep sampling. We have also used pheromone formulations to keep beetles in one location following implementation releases. Newly released beetles disperse rapidly and lose the critical mass needed for aggregation. The reason for this was revealed when Alex Gafke and his group showed that beetles kept in storage containers, like the ones shown below, cease pheromone production and are unable to form aggregations shortly after release. Providing artificial pheromone sources at release sites remedied that situation. As we will see later in this presentation, calling beetles to high priority targeted tamarisk is an important use and has been demonstrated in the work of Gafke et al. Keeping beetles away from stands of tamarisk with value as wildlife habitat while calling them into others deemed detrimental to riparian ecosystem quality is another use of semiochemicals. Dr. Cynthia Brown from Colorado State University, Fort Collins, and Hannah Erdl, now with the nonprofit Trees, Water, and People, approached us to help them test pheromone use within an IPM program to control tamarisk in the remote Mancos River Canyon, pictured in the upper right corner of the map. We had previously assisted in mapping tamarisk on the Ute Mountain Ute Reservation and were very interested in continuing to assist the tribe in controlling the shrub. The Mancos River flows through a remote and culturally significant part of the reservation. Unfortunately, it also has a large and persistent tamarisk infestation in need of control. One of the major problems with any form of tamarisk control is that the plants are resilient and will resprout after treatments designed to control them. If beetles could be focused on resprouting tamarisk, we could reduce control costs and herbicide usage. Cynthia Brown and Hannah Erdl designed experiments to test the use of semiochemicals as a component of tamarisk IPM, specifically targeting post-treatment tamarisk regrowth for defoliation by beetles. They cut or masticated tamarisk at sites on the Mancos during year 1. In year 2, Dr. Zeynep Assoy from Colorado Mesa University worked with a student from the CMU chemistry department, Lydia Dawson, to synthesize a batch of pheromone. We obtained GLV components off the shelf and formulated GLV baits. Brown and Erdl hung GLVs at 4 test sites early in the season and left 4 sites as controls. Later in the season, they hung pheromones at the test sites. They measured beetle population density as well as tamarisk growth and defoliation. The results were encouraging. Here is a summary of a poster presented by Brown and Erdl showing that semiochemicals attracted beetles to the area, seen in the top graph, and that beetles achieved higher densities on plants in treated plots, especially note July 28th and August 15th, the red bars. Defoliation was noted in experimental plots, but also seen in control plots. We believe that beetles were attracted to the area, but that 500 meters was insufficient to prevent beetles from spilling over from the experimental plots to the control plots. Most of the Mancos Canyon saw little or no defoliation that season, indicating that the semiochemicals had brought about the localized defoliation events seen that year. The lower graph shows a decrease in tamarisk canopy in treated plots over the course of the season. The overall results were encouraging enough to lead to a full-scale trial of semiochemical use within a tamarisk IPM program, and that will be conducted beginning in 2023. The southwestern willow flycatcher is a songbird known to nest in tamarisk, and in nesting territories where this happens, tamarisk becomes a protected shrub. On the other hand, tamarisk does diminish the overall value of flycatcher habitat when present as a monoculture or near monoculture. To work around this conflict between tamarisk as poor habitat and tamarisk as nesting substrate, preservation of some tamarisk stands combined with control of others is a good strategy for optimizing ecosystem services. Using the repellent to push Diabrabida away from areas where the flycatchers utilize tamarisk, and pheromones and GLVs to pull them to areas where the flycatchers do not utilize tamarisk, is a possible solution to the long-standing conflict. In conclusion, semiochemicals could be useful for manipulating populations of tamarisk beetles, as shown by Gafke et al. Semiochemicals can also be used in integrated weed management programs, as shown by Brown and Ertl. Semiochemicals may be useful for more complex population management, such as habitat preservation, where invasive plants may be providing transient ecosystem services. Other weed biocontrol systems could also benefit from the use of semiochemicals in the same ways listed above, and possibly in additional ways. Thanks to Bob Bartelt and Allard Cossé for a long-standing interest in semiochemical use in biocontrol, and for discovery of the pheromone blend. Thanks also to Alex Gafke, now with the USDA ARS, for his outstanding work showing the uses of semiochemicals in the implementation of biocontrol. Thank you, and if you have additional questions, you can reach me at my email address.